Dear Michaela, the floor is yours, and very nice to have you here.
Okay, thank you very much, Marco, and for this nice introduction. And it's a pleasure for me being here after the summer session and talking about maybe you think a very focused issue, which is the use of 3D models for study a specific endocrine tumor, which is the adrenocortical carcinoma. But I think that the concept that I would like to introduce and discuss with you in this talk would be also useful for being applied to different type of tumors, and not only the adrenal one. So just a brief introduction of our interest in my lab, and in particular in the context and microenvironment I'm working with in, which is the University of Florence. We have, the part dedicated to research, which is, in strict contact also with the clinical part, which is composed by two different hospitals, the Azienda Ospedaliera Universitaria Careggi, but also the Mayor, which is a specialized hospital for pediatric pathologists. So this is a unique, interactive, and very stimulating scientific environment where clinicians can talk and interact with preclinical scientists, which is really unique in my experience. And in this context, my group, actually I have two different groups, is interested in two aspects which you might say are totally different ones. So the mechanism actually working in the adipose tissue, in the adipocytes and in the stem cells, which is the interest of the adipose lab, trying to delineate the molecular and hormonal mechanism which are involved in the different pathologies, metabolic pathologies such as diabetes and obesity and metabolic syndrome and so on, by developing in vitro and ex vivo or cell and tissue models derived from human material. And the other aspect of interest, which is the adrenal lab, actually focused on the characterization and study of this specific endocrine tumor, which is the, it is composed by different type of tumors affecting the adrenal gland, which I will illustrate during the talk is a complex endocrine gland containing two different identities. So the type of adrenal tumors are very heterogeneous, very poor knowledge about the mechanism actually involved in this development. And again, our group is interested in development of cell and tissue models to study this peculiar endocrine type of tumors. Seems two different, issues, the adipose and the adrenal one, but they are not. When you think about the microenvironment where the tumor developed in, and in particular the adrenal develop in a microenvironment which is composed by a thick fat pad. So the adipose tissue can locally interact and modulate the development and progression of the tumor, in particular of this tumor, because the adrenal gland is surrounded by a fat pad, but also at distance. The adipose pads, the different adipose pad in the organism can secrete different hormones and regulate the distance, the development of the tumor. Since the adipose tissue is one of the main, I would say, endocrine organ in the organism. So it seems that there are totally different issues, but they are linked by the effect that the adipose tissue can have on the cancer. And more recently, since we are interested in developing different cell types, also not only from adult humans, but also from fetal humans, I collaborate with Marco and other friends, Sabrina Tait, and Miguel Sogorb, and we set up a consortium called Neonate, which actually was focused on development of different strategies for studying the developmental toxicity, not using animals, but using human cells, in particular from placenta and from human fetus material, to study in an in vitro organ on a chip the effects of different chemicals and insult altering the developmental structures of the embryo. So in this sense, we are interested in development different alternative type of system in order to study chemical toxicity by using cells, in vitro cells, and maybe also 3D spheroids derived from fetal human cells. So this is just an introduction of what we are interested in, and now I'm going to go through the real talk of today. And the goal of my talk is to convince you that in order to significantly step ahead, the research on adrenal cancer needs innovative, cutting edge in vitro 3D models, not only animal models and 2D models. And this why? For these two specific reasons in adrenal. Because the tumor should be addressed, taking into consideration not only the cancer cells, but also its microenvironment and its spatial complexity. And this is very important, in particular in the adrenal, because as I told you, in the adrenal, two different endocrine organs coexist. The cortical part with its steroidogenic activity, and also the medullary part more in the center of the structure, which produce catecholamine, epinephrine, and norepinephrine. So in this sense, it's really very important to study this type of complex organs in a 3D structure. And this last point of the coexistence of two different populations is a particularly relevant for the adrenal, because since the phylogenesis, during the phylogenesis of vertebrate, the coexistence of the two components are associated together only in mammalians, because in the different invertebrates and also other type of animals, the two acting glands are geographically separated. So it's important to have them together. And this is important also during the development of the embryo development of this organ. And since ontogenesis recapitulates phylogenesis, during the very early weeks of human development, cells from the neuroblast start a long journey migrating towards the organizing adrenal cortex up to the neoforming kidney, and migrates centripetally in this organ to give rise to the medulla secreting catecholamine. And to further support this unique plasticity of this endocrine organ, of this gland, in addition to the zonal articulation of the cortex, you can see here that there are different zones actually secreting different type of steroids. The mineral corticoids, the zona fasciculata actually secreting glucocorticoids, and the zona reticulata, which is more inner in the structure, which secrete steroid and androgens. This type of structure only in humans is replaced after birth by a different type of cells that actually constitute the adult adrenal. So there is a further complexity, not only the structure, the different structure of two different organs together, and also in the cortex, different zones producing different hormones, but also plasticity during the embryogenesis. And a strict interaction between this outer cortex, steroidogenic secreting, and the neuroendocrine compartments occur in the gland by a reciprocal regulation of the secretion. So the cortex regulates the secretion of the medulla, and conversely, the medulla can regulate the secretion, the steroid secretion of the cortex. By the secretion of different paracrine and also hormone secretion from the two parts. And even by direct interaction, contact between the chromaffin cells and the cortex. Chromaffin cells can also be found not only in the central part of the gland, but also dispersed in the cortical outer part of the gland. So there is really also a physical interaction between these two components. And this crosstalk peaks in regulation of coordinated responses resulting from the cooperation of these two adrenal components, such as the stress response initiated by the catecholamine, and then integrated and maintained by the cortical secretion, and also the inflammatory response. And therefore, integration between the cortex and medulla must be relevant not only in the physiological situation, but also during the development and progression of the cancer. These are the main characteristics of the adrenocortical carcinoma, ACC, which is a malignancy of the cortical part of the gland. It's a rare tumor, very aggressive, with poor prognosis, in particular when it is metastatic at the diagnosis, and only radical resection, also associated with mitoten therapy, is actually the only functional strategy for fighting this type of tumor. And conversely, the pheochromocytoma, which actually is the cancer affecting the medullary part, is often benign and is secreting catecholamine, so norepinephrine and epinephrine. But my talk of today is more focused on the mechanism and on the models that actually can be used to study, in particular, the adrenocortical carcinoma, which is the main focus of my group. So, which models? So we have in vivo and in vitro models, and during my presentation, I like to, to compare the different models, and in particular to stress the importance of using in vitro cell, combined models, in particular 3D models for study also the cancer, not only the physiology. And so starting from the typical model of the xenograft used, obtained by injecting a cell, cancer cell into the mouse. You can see here that we compare the different, aspects of this xenograft with the use of in vitro 2D models and also the possibility of co-culturing cell together in vitro, and also the possibility of developing spheroids and adenoids from the cells. And you can see here that if you compare the main important aspects to be studied in cancer, in this type of cancer, only adenoid, which are composed by both the cortical and the medullary part, can actually address these different aspects, which are the spatial organization, the possibility of study also the cells composing the microenvironment, so adipose cells, but also infiltrating immuno or cells, and only adenoids actually can study this interaction of the two component cortical and medullary one, which I hope that I'm really convincing you that are important for guiding the tumor. So we have, just to have a brief excursion of the typical models, animal models and cell models are used. We have the mouse xenograft with implanted human ACC cells. You can see here there are the different type of cells actually available from human. We have primary cells, in particular H295R cells, cell lines, which maintain the ability of this cancer to secrete steroids. But we also have lines obtained from metastatic cancer. All these type of cells can be used for be inserted in the nude mice, so immunodepleted nude mice, and you can obtain xenografts by injecting subcutaneously or also by putting them intravenous, or you can also have autotopic transplantation, putting the cells under the kidney capsule. Another type of mouse xenograft is the patient-derived xeno, and can be obtained by grafting not ACC cells from a cell line, but ACC fragments, so tissue from a patient, or ACC cells obtained from a specific patient. They're particularly useful to test the response of that specific tumor of that patient's therapy before using them on the subject itself. However, when screening the efficacy of the immunosystem therapies in this type of patients using this personalized patient-derived xenograft, which the immunosystem therapies actually the immune response of the patient, this type of models, which have been obtained in nude mouse, are not useful because you don't have the immunocompetent cells, because in order to maintain the graft from human, these are immunodepleted, the mice system. And for this specific type of screening, so analyzing the immunocomponent response to the tumor and the immunotherapy, the model has been implemented by providing the mouse of the humanized immune system derived from the patient. So we can deplete the mouse from its own immune system and obtain and collect the immune system from the patient and observe the response of the immune system of the patient in the xenograft obtained from, using the tumor tissue. So they are very complicated but very informative. In particular, when we want to test this new strategy of anti-cancer therapies. And this is an example of a humanized ACC mouse model where the immune checkpoint inhibitory therapy with pembrolizumab was tested with very good results on the xenograft growth here before using it in the patient. So the tumor has been implanted in the humanized animal, and the pembrolizumab used to follow the growth of the tumor. And then, since the tumor in vitro was non in vitro, in the mouse was responding to the immunotherapy, it was used also on the patient, and you can see here obtaining very good results since there was a reduction in the progression of the tumor and increase in cytotoxic CD8 T lymphocytes infiltrating, in particular, the liver where you have the metastasis. To further assess the role of the local tumor microenvironment in controlling the tumor progression through 3D interaction of the cells, both tumor explants co-culture with additional microenvironment cells or 3D spheroids generated from tumor cells or alone or in co-culture with other cells derived from the microenvironment has been used. So you can add different type of cells in the systems to study the interaction with the cell microenvironment. And a further step in this type of 3D technology, cell culture, to better reproduce the tumor microenvironment interaction is also to move from the static condition of putting the cells or the tumor fragment in the matrix and the wells and studying the effects of or drugs or interaction with other cells is applying to this system a microfluidic system, which actually better resemble the vascularization situation condition of the tumor mass. So we can use this different type of approaches, cellular tissue fragments and also spheroids, but putting them in a microfluidic system, which is really more physiological than just putting the drugs in the in vitro in the wells. Here you can see the generation of ACC spheroids from H295R cell line, so the primary ACC tumor, to assess in vitro the effect of on apoptosis of a combination of mitotain, which is actually the current drugs chemotherapy used in ACC, and also the tyrosine kinase inhibitor sunitinib. You can see here that when you put together this two drugs, you can stimulate apoptosis in this organoid spheroids, not organoid, derived from H295R cells. But actually, this type of organoid has not been so well characterized in this work. So the next type of in vitro 3D system has been derived and developed by Constance Hantel in Zurich, one of the major specialists in cell and animal models for ACC study. And actually, she elegantly developed an alternative 24 wells technology for producing these spheroids starting from different cell lines. You can see the primary ACC H295R cells, and also the cell line derived from the metastatic ACC. You can see here that she derived high number of spheroids, not only very few ones. So this system is a sort of high throughput system for generating spheroids in vitro. You can see here that she studied the proliferation by Ki67 staining, and also the expression of a steroidogenic enzyme, such this one, in order to test to assess the functionality of these spheroids. SF1 is the steroidogenic factor 1, which actually is a marker of the cortical origin of these cells. However, these 3D spheroids are composed only by cortical cells and lack the physiological condition of interaction with the medullary component. To reproduce in vitro, the integration between these two parts of the adrenal, which we have seen, I hope that I have convinced you that so relevant for the functionality of the gland in physiology, but also in cancer. A group actually generates an organoid from an explant of a fetal adrenal, including this specific fragment obtained from fetal adrenals, and culturing in vitro, studying the evolution of this ex vivo system for some days. You can see here that they have the cortical part here. This is the marker of the cortical steroidogenic part, but also the medullary surviving during the culturing. And during culturing, they only measure the activity of the cortical part. So they measure androgens and glucocortical secretion after culturing in vitro, but they say nothing about the medullary component. So we actually don't know if during the culturing for some days, this structure really maintained the both aspect of the adrenal. So our group in Florence started with a different strategy and approach, developing a 3D in vitro organoid model of fetal adrenal from adrenal primary mixed cell population. So we have cortical and medullary, obtained by legal voluntary abortion between 9 and 12 weeks of gestation. We perform in our university hospital. So from the aborted material of 10-15 fetuses, we derived a mixed primary cell culture. You can see here the morphology, so are both medullary and cortical cells. And we characterize the presence of the two parts, and by measuring markers of the cortical part and markers of the medullary part, chromaffin markers, and also measure the ability of these cells to produce cortisol in basal condition, and this depends on different gestational age, but also after stimulation with the ACTH. So this population, our mixed population, they can respond by secreting hormones. And hope that is work okay. And after 10 days of in vitro culture of this mixed population, by lowering the serum condition of the culture, the cells, which you can see display a great mobility, spontaneously aggregate, giving rise to these 3D spheroids, which slowly detach from the plastic well and keep on growing for additional 10-15 days. And we call these spheroids adrenoids. And double immunofluorescence analysis of this CYP17A1, which is a marker of the cortical part, and tyrosine hydroxylase in red, which is the marker of the medulla, show a similar cell distribution and also a similar 3D architecture in adenoids derived from 11-week fetal adrenals, which was very similar to the structure and the staining of the 11-week fetal adrena the adrenoid was derived from. So suggesting that this forming organoid actually maintain the functionality, but also the structure, the architecture of the fetal adrena itself. Well, another important in vitro system to investigating, in particular, tumor microenvironment is in the in vitro cell culture. So we looked at the possibility of, investigating using this 3D system obtained from fetal. But another important model is to look at the ability of the cells in interacting with other components of the microenvironment. And I told you that our group is really interested in the adipose part. And you can see here that in particular in the tumor in advanced stage, in adrenocortical tumor in advanced stage, there is a disruption of the capsule, and the cancer mass is infiltrating the fat surrounding the adrenal. So putting the cells in contact, the tumor cells in contact directly with the adipose cells. So there is an interaction, and we can reproduce we can reproduce this interaction in vitro by using the cell lines, the H295R cells, and culturing them in this specific wells where there is a sort of separator of the two population with adipose stem cells derived from human subjects, so we can study in vitro the interaction between these two populations. And we found that in this condition, the tumor cells inhibited in vitro stimulated adipose differentiation from this ASC, as you can see from the decrease in this marker adiponectin, BP4, and hormone-sensitive lipases, which are markers of the mature adipocytes. And when the ASC stem cells were differentiated towards adipocyte, which is indicated by the bar marked adipo, in the presence of the tumor cells, you can see here that there is a decrease of the differentiation of these cells, also associated with a decrease in the number of lipids droplets actually inside the differentiating cells. And on the other cells, so on the other hand, there is there is an effect of the adipose cells on the tumor cells, because these undifferentiated adipose cells, the stem cells, actually stimulate the invasiveness and transendothelial migration of the tumor cells when they are culturing together. So there is a reciprocal interaction between the adipose components and the tumor cells. Finally, the development of 3D tumor spheroids containing cells from the microenvironment would be the best model to assess this interaction, the tumor response to the microenvironment, and also to chemotherapies that actually are not impacting directly on the tumor, but on the tumor microenvironment. So this is very important because the microenvironment is a very heterogeneous population containing adipose stem cells, adipocytes, immunocompetent cells, and this microenvironment can really modulate the tumor progression and be the target of novel chemotherapy. So we can study this interaction with, cells derived from the microenvironment in creating specific organoids, specific spheroids. So three-dimensional structure where you can see the different type of microenvironment cells interacting in a 3D structure, which is really important for guiding the response of the tumor. As you can see here, a paper where an in vitro spheroid obtained from the neuroblastoma cell line SNAS were infiltrated with fibroblasts, which you can see here can organize spontaneously surrounding the tumor SNAS mass, so giving rise to mixed infiltrated organoids with is really interesting in for testing the effect of chemotherapies directed not only against the tumor but also towards the tumor microenvironment. And you can see that this mixed organoids, this spheroids, has been used for studying the effects of a combination of drugs directed against the tumor, the vincristine, but also against the stroma. So the prostaglandin inhibitors, you can see here that when they are combined using something that which is directed against the microenvironment, you can see a reduction in the proliferation of the spheroids. So again, so this is my last slides. We developed a 3D model for studying the cancer, which actually contains both type of adrenocortical cells and medullary cells. So this organoid, we call mixed adenoids, has been developed in vitro, starting from cell lines of cortical carcinoma and also pheochromocytoma. And we mixed together, and you can see here, you can follow the growth in vitro of this mass, these spheroids, in the lower part obtained by using only MTT, which are derived from the few cell lines. And here you can see the growing of the mass when you obtain the spheroids, the adenoids, by mixing the adrenocortical carcinoma cell lines and also the few cell lines. You can see here that the mass is actually growing better, and it is more defined compared to the mono spheroids obtained only from culturing the few cells. And you can see here that we assess also the reciprocal component, cortical and medullary, during the growing of these spheroids. You can see here that we compare the monospheroid growth and the mixed spheroid growth, but also check for the presence of specific markers of the cortical and medullary part. You can see here that during the culturing for 10 days, you have a modulation on the expression of the markers of the cortical part and the marker of the medullary part with an increase in the medullary cell proliferation. But you can have both type together. So maintaining the crosstalk between the two components of the gland, which actually can influence the development of the tumor and also the response to the different drugs. So all these type of models, in particular the 3D models, are really important for the study of different ability of the tumor cells to behavior in the growing mass, and they are really functional to assess the functional activity of the adenoids. And obviously we have to further investigate this ability of the adenoids to maintain the respective function of the cortical and medullary part. And they can be used for generating mixed spheroids after engineering the cells. So we can modify the cells by inserting specific mutations that are important for the development of the tumor. And they are really very important to study the interaction with the microenvironment cells, adipose cells, and immunocompetent cells. So in conclusion, I hope that during my long talk, I have convinced you of the necessity of developing novel 3D cell models, integrating cortical and medullary components and tumor microenvironment cells to better study the response of the adrenal tumors to novel anti-cancer therapies. This is not a focus and limited to adrenal cancer only, but this type of a mixed system in 3D structure is really important for the study also of different types of cancer. So to respect the composition and the heterogeneity of the tumor microenvironment and of the type of tumors, but also to respect the 3D structure, which can actually be very important also as a target of anti-cancer therapy. So I thank you for your patience, for your attention, and I thank all the collaborators in my group and also external collaborators, and obviously also the funding agency that we have for this type of study. Thank you again. I'm ready for your question.